Okay, so we are looking at C2, which is going to include both the chemical signaling component, which is HL only, and then in the second half of the video, we'll do the neural signaling, which is everybody. So if you are standard level, fast forward a little bit towards the later part where I erase the board and write C2.2 and pick up with us then. So this first component of chemical signaling is HL only. So I'm just gonna make that clear from the beginning. And this is something that in the old course was kind of spread throughout, and in the new course they have kind of consolidated it as a single concept. So instead of only learning about hormones with endocrine and neurotransmitters with neurons and calcium in terms of kind of muscle, now they have kind of combined them into one concept, which is to understand the idea that chemical signals are released from one cell to give instructions to another cell. So that's what we're meaning when we're saying chemical signals, is basically how do cells communicate with one another. So I'm going to go through the three kind of steps that are involved in this process. So step 1, 2, 3, and then we're just essentially going to work through kind of 6 examples that you need to know, because it looks like most likely if you're asked about it, will be in the context of one of these examples. So if we are talking about the general process involved, can be summarized as a 3-step process. So step 1, and these are the steps from essentially release. So step 1 is going to be reception. Change to purples. Step 2 then is a signaling transduction pathway. So step 2 is the transduction pathway. So signal transduction pathway. And these can be fairly complex. So we will look at a few examples of these. And some of them you need to know a little bit more about, and some you do not need to know in as much detail. And then the third step, step 3 then, is going to be response. So then step 3 is response. I'm just going to try turning out the lights closest to the board. See if that reduces my shadow. It might just be further down, but I think it looks better so far. Okay, so those are the 3 steps that we're going to need to talk about. I want to do a little bit more on step 1. So step 1, when we say reception, what we mean is that the actual chemical signal, which we are going to call a ligand. So that is a vocab word we need to know. So the ligand, which is the chemical messenger, is going to bind to a receptor protein. So the receptor is always a protein. Okay, so that's our kind of extra information, and we're going to spend a little bit more on this step and being clear about types of ligands and types of receptors. The step 2 and 3, they're quite diverse depending on the pathway. So I'm not going to go into specifics on step 2 and 3 until we get to the actual examples. So when we go through the examples, we'll look at what does the pathway and response look like for that example, but we won't go into as many things kind of first. Okay, so when we look at ligands, you do need to know a few examples. So there's 4 examples that they essentially named as examples of ligands, and we're definitely going to spend more time on 2 of them. So we'll cover the others just as examples, and then we'll really zoom in on the other 2. So some of the examples they included was that calcium ions can act as a ligand, and we have actually referred to these a couple different places. So they are active, they play a role in our neurons, which we will cover in part 2 of this video. They also play a role in our muscle cells. So they are actually what's going to be responsible for making the actin heads available, or actin sites available to the myosin heads. So they play a role there as well in terms of acting as ligands or signals. You do not have to know any specific examples, just that it is a type of ligand and is obviously a single charged ion. So that's really the reason for it, is just to recognize it's quite chemically unique by doing that. The next example that is named, but again, you don't have to know any specific pathway for, is cytokines. So we talked about those a little bit in the immune system. So they're going to play a role in kind of immune cell activation. So we did talk about their role in terms of helper T cell to activated B cell, kind of really initiating the adaptive response. So we're just going to put immune system here. And there's lots of different cytokines that play a lot of different roles. Again, there's no specific ones you have to know, but it would be worth, if you're reading a question about cytokines, to recognize that they are, of course, an example of a ligand. The next two are the ones you need to know quite a lot about. So neurotransmitters are ligands. And so we are going to talk quite a bit more about those in the second half, but we'll talk a little bit more about it here as well. And so when we talk about neurotransmitters, they are involved in what is called local signaling. Cytokines and calcium usually are as well. What that means is they tend to be signaling to cells that are right near it. So when we look at neurotransmitters, we know that's right across a synapse. And generally cytokines, if we look at the example with like helper T cells and activated B cells, they have to literally be in contact with one another. So these are kind of signals that go directly where the two cells are. They need to be close to one another. The most unique then in terms of the different pathway would be hormones. And we're going to include several examples of hormones. So we're going to talk more about those. And those are going to be involved in distant signaling. So as we already know from doing homeostasis stuff and endocrine, those travel through the bloodstream. So they're able to be released from one cell and actually activate a farther removed cell, so they can transmit messages across some distance. For just particularly these two, want to give you a little bit more about what kind of chemical background they could have, so what they could be actually made up of. So neurotransmitters are quite diverse. So they can be made up of single amino acids. They can actually be made of like smaller amines, which are even smaller kind of components of that. They can be peptides, which are just going to be really, really small protein chains. So neurotransmitters are generally quite small, would be what I would say is they're generally associated with several kind of amino acids. Nitrous oxide acts kind of as a similar one. And so that was named in the course as well. Nitrous oxide is what we give for laughing gas. So that's another example that acts on neurons. So it acts as a ligand in that same category. Hormones tend to be larger, so you can see some they're rarely single amino acids. They tend to be slightly larger amines, peptides, proteins. And then the other one that we need to add is that they can also be lipid-based. So probably the main thing we want to see is that we do actually have hormones that can be lipid-based, nonpolar, uncharged. And also that generally speaking, hormones are larger than neurotransmitters. But they're all kind of quite diverse. So part of the thing that you need to recognize is that there's not one type of ligand. So we do have single ions, we have large lipid-based hormones, we have larger peptide chain neurotransmitters. So there's a lot of chemical diversity, and that just enables them to activate really unique receptors and activate a range of different pathways. So lots of diversity here enables more different kind of types of communication pathways. Okay, next thing we want to talk a little bit more about is the receptors. So when we talk about receptor proteins, there are two main categories. So what they have in common is they are all proteins. But there's two different kind of locations they can be, and we'll pick this up in our table as well, and for each one identify what type it is. So they can be intracellular, which means basically that they're in the cytoplasm. Some examples where they would be all the way in the nucleus, but generally they're going to be in the aqueous cytoplasm. And because the cytoplasm is aqueous, the actual receptor needs to be hydrophilic. So the receptor itself has to be able to remain dissolved in the cytoplasm. Interestingly, the ligands that bind to it are actually the opposite. So it is hydrophilic, and this is only used for non-polar ligands. Because in order for it to get into the cell, it will need to cross the cell membrane, which it can only do if it's nonpolar. So hydrophilic protein, nonpolar ligand. The other option is that it's sitting as an integral protein inside the membrane. We call these in this context transmembrane. And they are going to sit through the whole membrane. So as a result, they have to be amphipathic. So they are going to have hydrophilic and hydrophobic regions. And in particular, they have to have a decently large hydrophobic pore to create a passageway in the membrane. So they have kind of a central part of it that is essentially made up of hydrophobic amino acids that sit, and then they create a pore. So hydrophobic is what creates the pore. So I want to be clear that the hydrophobic is where the pore is. The pore itself becomes hydrophilic, so they kind of wrap around. A little bit more complexity than you need, but you have to know that they do need both regions in order to sit in there. And these are really good for charged or polar ligand. And so this is for ligands that cannot cross the membrane, so they bind at that actual receptor. So that's kind of fairly brief, but that's some of the background. The rest I want to cover through the examples. So again, when we look at pathways and responses, there is so much diversity that the best thing to do is just look at the 6 examples and say, what's the pathway here? What's the response here? So the rest of your page is going to be one giant table. So we'll set it up, but I would not do your 6 rows yet because they are going to be different. Like the G protein one needs to be quite a bit bigger. So set up your columns across and then we'll just fill in as we go. So we're going to look at the 6 different named ligands that you need to know about and what you need to know about their pathway. So I'm just going to erase all of this so we have space. So it's always within possibility that they could present a new signaling pathway to you that you haven't learned about and ask you to interpret from a diagram. But these are the ones that you could have to be asked to explain like without provided extra information, that the expectation is you know some stuff about. Okay, so for our table, we're going to start with what is the ligand. Then we're going to say what kind of receptor does it use. Then we're going to summarize based on how much they expect you to know. What does the transduction pathway look like? And that varies a little bit. In some cases, we're just going to put like series of enzymes because you're not expected to know details. In a couple cases, you do need to know a few of the first steps. And then what is the response? And when we do the response, we're going to practice working on kind of the immediate response and then also the kind of zoomed out response. So what is the cellular response? What is the impact on the organism? So trying to kind of trace both of those. I'm going to move this over slightly just so I'm using more of my space. Okay, the first thing that is specifically named is a pretty small example, but the first one is that they do want you to know about this example of it being used in bacteria. Bacteria, of course, are interesting because they're not multicellular. So this is, in normal cases, in most contexts, we are talking about cells within an organism communicating, but this is actually an example where unicellular organisms are communicating with each other using this process. So this is the ligand that you need to know about are called autoinducers. So that is what we call the actual name of the ligand. I want to add a couple more things here. So if you were to see autoinducers, I want you to know that those are in bacteria. So this is kind of our unique example. All the others are going to be things we would see in us. The other thing you might see is not see the term autoinducer, but you might see it referred to as quorum sensing. So quorum sensing is essentially the process that uses autoinducers. So just making sure we would recognize either of those terms. Okay, what is the receptor? So it is an intracellular receptor. You don't have to know the specific name of it, just the general idea of it. So we call it the Lux protein for bioluminescence. But any of these are always going to operate by binding to an intracellular receptor. And what happens is the actual autoinducer only enters the cell at high concentrations. So it diffuses into the cytoplasm when it's in high concentration. So if a bacteria is lone, if there's only a few, they release autoinducer, and then because there's not many near them, autoinducers remain in a low concentration, so they don't reabsorb them. So they release it, and then it just diffuses out. If, however, there's a bunch of bacteria colonies really close together, and they're all releasing autoinducers, then the concentration of autoinducers goes up, and then it travels from that high concentration back into the cell. So the cell both releases and absorbs, in which case it will absorb it. When it is actually re-entered into the cell, it will actually bind to a protein receptor. We're going to look at the specific example bioluminescence, and it is called a Lux protein receptor. So it's going to bind to the Lux protein receptor, and then it is going to activate transcription. So I don't think you probably need to know the specific name of the Lux protein. The key is that it's going to act in this role of binding to the DNA and starting transcription. Okay, so like I said, we're going to look at basically what are the response at a cellular level, what is the response at an organism, or in this case, colony level. So the response at a cellular level is gene expression. So what's happening is the cell is now expressing the gene that creates the bioluminescence. And if enough cells are doing it and expressing that, it creates the fluorescing protein. And then that fluorescing protein emits light, and the result at a colony level is bioluminescence. This can also be done for biofilms. So there's other uses for it as well, and you don't have to know it specifically for bioluminescence, but you could have to give an example. So the whole colony, if they're all expressing this protein, the result overall is the glowing effect of bioluminescence. Okay, so that is our first example that we need to know just that little bit about. The next one I'm going to do is acetylcholine for the sake of this topic, but we are going to do acetylcholine again in the second part. So we're going to do a pretty small version of it right now, and then we'll talk more about it. So acetylcholine is a neurotransmitter. And it's come up several times, so it is also the one that you're going to have to know about in neurotransmission. We also talked about it in the context of neuromuscular junctions and the activity of sarcomeres. So again, anytime something shows up a couple times, you're like, oh, they want us to know about this. Okay, it is a transmembrane receptor. Transmission. So it's a transmembrane receptor, and it is going to be found on postsynaptic dendrites. And so we're going to explain what's happening at the receptor level here in terms of how it leads to these changes, and then we'll talk more about what that does to the neuron in the second part. So here's going to be my receptor. The key thing I want you to know here is that it facilitates a shape change. So this would be an indication of it, and that would be what it looks like when there is nothing bound to it. So that's sitting in the membrane. When the acetylcholine binds to it, it leads to this shape change. And what that's going to do is it's going to cause it to separate, and it creates that pore, like so. And again, that's only going to happen, I've got too many markers in my hands, that is only going to happen when acetylcholine is bound to it. And that is going to create an ion channel. And that ion channel is going to allow sodium to enter the neuron. So if we want to kind of quickly summarize that, we want to make sure, what do we know that has happened? Is that there's been a shape change. So there's a change in shape. The result is that it creates an ion channel. In the protein itself, and that is going to allow sodium to enter. So that's kind of the detail you need to know about the pathway. Shape change, creates a channel, allows sodium in. So let's look at what is the response. So the immediate, of course, response is this sodium influx. What that actually causes is depolarization, which we'll revise again what that means next time. Depolarization is going to cause the action potential to be spread. And then we do want to have a specific note of what does that actually look like for acetylcholine. So we remember that acetylcholine is involved in neuromuscular junctions. So the ultimate kind of bodily impact is going to be muscle contraction. So for these responses, we're doing cellular to organism. So the cellular impact is depolarization leads to action potential propagation. What happens to the organism? The organism is able to make a muscle contraction. Okay, so I'm going to just keep my table here, but erase because the next one is the G protein one, so we need a little bit more space. All right. So we're going to talk, so it is specifically in there that you need to know about G proteins. So rather than doing it separately, we're going to do it through the example of epinephrine. So epinephrine is going to be our ligand. Epinephrine is a polar hormone. We are talking about it in the context of hormones. Epinephrine can act as a neurotransmitter as well, so it is a small kind of peptide hormone with charges. We are talking about it in its context of having moved from the bloodstream to liver cells, so we're looking at its capacity as an actual hormone. So that is going to be my primary ligand. I am going to add one more thing, that this pathway also involves a secondary messenger. So after epinephrine, it then has a secondary messenger, which is a concept we need to understand. So the secondary messenger we don't generally call the ligand because it has already relied on a different message starting the process, but usually it then moves in the cytoplasm. So this is kind of the intracellular messenger compared to the outer one. The specific example is cyclic AMP, and you do need to know cyclic AMP is a common secondary messenger. Okay, let's look at the receptor. So this is a very special type of transmembrane receptor that you need to know about, which is called a G protein. So it is a G protein transmembrane receptor. And we are going to specifically look at the example of in liver cells. So epinephrine acts in a lot of places, but the example that we're going to look at the pathway is what it's doing in your liver cells. So I'm just going to make a note of that we are looking at a very specific context. So what I want to do is I want to draw what does the actual G protein look like at baseline, and then in the pathway we'll show what's actually happening there. So here we're going to have our transmembrane receptor, like this. So the reason why it's called a G protein is that it has bound to it this cluster of proteins. So it has kind of a large G protein, and then it has these kind of smaller subunits that are attached to it that are actually going to prevent the movement of the G protein. So it's got this G cluster. You just really need to know about the G protein. You don't need to know about like the alpha and beta subunits, as long as you understand it's this big G protein with that attached. Additionally, attached to it is a GDP molecule. And when the GDP molecule is attached, it is immobilized. So this is an immobilized protein unit. So it is not going anywhere. So that is kind of the nature of the receptor at baseline. So now we're going to look at what happens when it's activated. So we'll draw a mini diagram of that and then kind of clarify the key steps. I'm not going to go into the names of the specific enzymes. We did talk about those in class. They're not named in the syllabus. So I would not recommend worrying too much about memorizing those and focus on ones that are kind of very specifically have been given named examples so that you're not trying to memorize things you don't need to. Okay, so we have our G protein receptor. And then this example involves a second membrane protein. So this has a second protein further down the membrane. So that is kind of what's unique about this. This second one is called adenyl cyclase. And its job is going to be to activate cyclic AMP. So if you can remember, cyclase is in charge of the cyclic AMP. So this is currently inactive. So what needs to happen is my epinephrine is going to bind. So we'll have some epinephrine bind. So here comes my epinephrine. And what that's going to do is that's going to activate my receptor. At the time of activation, the GDP actually leaves, it detaches, and becomes energized to become GTP. That actually enables this G protein to break off. So its little subunits kind of stick around, but the G protein is going to move over here. So my G protein is going to move from the actual receptor over to the adenyl cyclase with the GTP as well. So the GTP plus the G protein activate adenyl cyclase. When they arrive at adenyl cyclase, that activates the adenyl cyclase, and when activated, it is going to produce cyclic AMP. So that's a kind of somewhat simplified version of it. I was trying to get away from the light, but anyway, it's right over it. So there's the receptor protein. When it is activated, the GDP detaches, allowing the G protein plus a GTP to move to the adenyl cyclase. At the adenyl cyclase, it is going to be activated and it is going to make cyclic AMP. Then cyclic AMP is going to activate a series of enzymes. So if we want to summarize that really quickly, so the activated receptor allows for GDP to detach, the G protein and GTP. So once it detaches, it becomes phosphorylated into GTP. So DP2 phosphates, GTP3 phosphates. GTP is like ATP. It's now an energy source. So G protein and GTP activate, move to and activate adenyl cyclase, which is going to make cyclic AMP. Cyclic AMP then is going to activate a series of enzymes. So there are names for each one. It's like protein kinase becomes protein phosphorylase, becomes glycogen phosphorylase. So there is a set pathway. Like I said, they're not named, so I think you're fine with just a series of enzymes in the cytoplasm. Okay, so that's kind of our steps involved. I'm just going to kind of match them to the colors in my diagram. So this is the activated receptor that is going to allow for the GDP to detach and become phosphorylated into GTP. That is what enables the G protein to actually move. That's going to come over here and bind to adenyl cyclase, which is my enzyme. You know it's an enzyme because it ends in ACE. It is going to then make cyclic AMP, and that is going to set off this series of enzymes, one of which activates the other. Okay, the last kind of enzyme to be activated is called glycogen phosphorylase. So I am going to include that one as kind of the response. So it's going to be activation of glycogen phosphorylase. When glycogen phosphorylase is activated, it breaks down glycogen into glucose. So hydrolysis, which is just breaking down of a polymer of glycogen into glucose. And so the result of that is going to be more glucose. More glucose is more ATP. More ATP facilitates the fight or flight response. So we'll just write ATP for flight or flight response. So we just want to remember that that is the overall reaction of epinephrine. And remember, you can call it adrenaline as well. There is like a little NOS point in your syllabus about how they mean the same thing. They're just different communities calling it different things. So if you see adrenaline, you would recognize it is the same thing. That's probably the pathway that you need to know in the most detail, because you do need to know a bit of this information here in terms of what's actually going on in the pathway. Okay, I'm just going to keep erasing them one at a time, so I'm not having to write tiny until the next couple we'll do together. All right, so number 4, so we've done autoinducers, we've done acetylcholine, we've done epinephrine, and we're going to do one more transmembrane one. So the next transmembrane one that we're going to look at is insulin. And again, I always remind my class, if you're seeing something show up a lot, it's a good candidate for like an extended part question. So there's a lot that we've done on insulin. We know about its structure. We know about its release in the endocrine system. We know about its impact on homeostasis in terms of diabetes. So it's everywhere. So let's make sure we know it. So I'm going to put insulin. And we're going to remind ourselves that this is also a polar hormone. So it is not going to enter our cells. The other thing I want you to make a note of here, just to remind you, because I don't think we need this to be a big separate point, but this is an example that is operated by negative feedback. So one of the last dot points in the syllabus is just to recognize that ligands can be regulated by positive or negative feedback. You shouldn't need to learn any new ones. You can just remember ones you know. Insulin's a great one to do for negative feedback. For positive feedback, you can try to do estradiol, or you can just do oxytocin is what I would do. That's easier. So as an example of a hormone, which is a ligand that operates under positive feedback. Okay, so this is going to be an enzymatic receptor. So it has a specific name, which is a tyrosine kinase. So ACE means enzymatic. Transmembrane receptor. Okay, so what I want to do is I want to draw again, kind of like I did on the last one, because this is another one we have to know a little bit of detail about what actually happens. I want to draw what it looks like before it's activated, so we can then show what happens when it activates. So it is going to sit like this, and it's going to basically have two receptor domains. Like so. And then when it is set like that, essentially these two intracellular components are inactive. So they are not energized. They cannot actually function as an enzyme at that time. So that's kind of the normal appearance of the tyrosine kinase one. So then let's look at what actually happens then when insulin binds. So when insulin binds, it particularly happens at the inside region. I'm not sure how great I'm going to be at drawing that is only happening at one region, but essentially it comes together. So it pulls these two regions together inside. So the two components when insulin binds actually join together. So here's my insulin, here's my insulin. What that actually does is it causes what's called autophosphorylation. So when these come together, they get phosphorylated. So each of these is a little phosphate group. And they come over and they bind there. And this is now an active enzyme. So that active enzyme is now ready to set a series of kind of protein or enzyme reactions together. I'm going to include the response here just because it happens at the membrane. So I think it's worth kind of looking at. So then as this happens, what it's going to ultimately do is it's going to cause a little vesicle over here to come and merge. And inside my vesicle is a GLUT4 protein channel. So it's causing that to move over to the membrane. So it's migrating a protein channel to the membrane, and that is a GLUT4, which is going to be a glucose transport protein. Okay, so again, let's write a little description of that in words as well. So when insulin binds, so the binding of the insulin actually moves and then activates the intracellular enzyme. So it moves and activates the intracellular enzymatic domains. It's called autophosphorylation, which basically means that when they pull together, the phosphorus groups just come to it. So they basically bind on their own. So it phosphorylates that area. The result of that is that is then going to activate an enzyme pathway. So again, it doesn't even tell us the names of the enzymes. Just one enzyme activates another, activates another. So then what is the response? So the response is going to be the movement of GLUT4 glucose transporter vesicles to the membrane. So the vesicle containing that transporter is going to move to the actual membrane. Now it has the ability to transport glucose. So glucose can be transported into cell. So the glucose can be transported into the cell, and then the glucose goes into the cell and out of the bloodstream. So the overall impact is going to be decrease in blood sugar levels or blood glucose. So we already knew that that is the impact of insulin. Now we can account for how does it do that. So if we were kind of writing about insulin generally, we've got a little bit more detail for that. All right, the last two are both intracellular, and so I'm actually going to do part of them together. So I'm going to do slightly a bit separate, but I'm going to actually do their picture together because they essentially work the same way in terms of the intracellular pathway. So we'll combine them a little bit. So we'll do two of them kind of together. And these are, again, things that we've done before. So you can see there's a lot of really nice cycling of things in this course. So it's coming back to things we already know about and just helping us understand it better at a cellular level instead of at kind of the bigger organism level. So there's two different ones, but they're very similar. So we're going to do O-estradiol. So remember, it is an international course. You can put the O or not put the O. You can call it estrogen, and they will give you credit for it. Although in this context, I do actually like the estradiol because we are looking very specifically at the form of it that is involved in the menstrual cycle, and some other forms of estrogen actually work on different receptor cells. So this is actually a better way of putting it. So that's why I'm going to leave it like that. And then the other one that you're going to do is progesterone. So this is a steroid hormone. So again, steroid hormones are lipid-based. This is also a steroid hormone. S is testosterone. Because we know so much about the menstrual cycle, we will stick with that and use that as our main one. Okay, so then we are going to say, what are the receptors? So in both cases, we are looking at an intracellular receptor. But what I want to make sure to add is what specific target cell we're looking at for the response, because both of these can operate on many different cells. So we want to be careful that a single hormone can activate different receptor cells. So in particular for estradiol, we're going to look at receptors in the hypothalamus. So we're going to look at receptors in the hypothalamus cells. And then for progesterone, we are going to look at receptors in the uterine wall cells. So again, particularly if you look at progesterone, it can also travel to the actual hypothalamus and pituitary and work through negative feedback there. So again, it has other ones, but these are the ones for this topic that you need to know. Okay, so then we're going to draw this one diagram for both, and then we'll cover their responses separately. If you haven't drawn your vertical lines, after this you can go back and draw vertical lines if you want. So again, I'm going to draw a single way because they work essentially the same way, and most intracellular ones work the same way. So there's my cell membrane. Here's my steroid hormone. And then because of the fact that it is steroid-based, therefore lipid-based, therefore nonpolar, its receptor actually sits inside the cytoplasm. Okay. There's some DNA. So what's going to happen is the hormone is going to come into the actual cell. It is going to bind to the actual receptor, and it's going to form what's called a complex. So a hormone receptor complex or a signal receptor complex. So it is going to physically attach to it. And instead of just activating it, is now going to move with it. So this is a hormone receptor complex. That hormone receptor complex is now going to move into the nucleus via one of the pores, and it is going to come over here, and it is now going to act as a transcription factor. So it is going to bind to a promoter to start the production of mRNA. So it is going to prompt this cell to now produce the mRNA. All right, so that's the main idea. Crosses the membrane, forms the complex, enters the nucleus. So again, we'll just do a quick description of that as well and say that the signal receptor or hormone receptor, you can call it either one. I've seen both terms, so I'm just using it interchangeably. Enters the nucleus and acts as a transcription factor. So enters the nucleus and acts as a transcription factor. All right, so we will do the responses separately. So in both cases, they are going to be transcription. We'll be a little bit more specific. So transcription of, and this is where you have to link it back to your menstrual cycle stuff, of GnRH mRNA. So that's your gonadotropin releasing hormone. That is then going to activate the pituitary gland, which is going to activate FSH release, which is going to lead to egg maturation. So it'll be kind of follicle growth. By FSH. So I've skipped a couple steps there, but you can go back over that in the context of your D3.1 reproduction stuff. But again, the first step is to make the GnRH. mRNA gets turned into GnRH. That goes down to the pituitary. The pituitary releases FSH and then LH. That leads to kind of follicle growth and ultimately ovulation when the LH is released. Okay, so that's kind of the pathway. So here we are also going to have transcription in progesterone, but it is actually going to transcribe a growth factor protein. So it's the mRNA for a growth factor. Once that growth factor is made, that is going to stimulate cell proliferation. And the stimulation of cell proliferation is going to create the wall thickening and also its vascularization. So the way to get a thickened wall is for the cells in there to undergo mitosis quickly and create more cells. So that is kind of the specifics of how do you go from progesterone to the thickened wall. It's just adding that extra step in. Okay, that's everything C2.1. C2.2 is next. Okay, so we're looking now at neural signaling, and this one is a lot of SL as well. In fact, the big difference is HL largely just has to add some terminology. So conceptually, SLs need to know quite a bit of it, but you can get away with using less terms than HL needs. So what I want to start with is just kind of looking a little bit at the idea of there being both electrical and chemical signaling involved in this process. So I want to show kind of two neurons, and we'll use that to label neuron structure as well, and look at the idea of them being two different kinds of signaling involved in this overall process. So I'm going to put this kind of right under our title. So I'm going to draw two neurons. So here we have our first neuron. This is going to be the actual cell body, which is like the soma. Then we're going to draw an axon, and then we'll draw some axon terminals with some terminal buttons. And then we're going to draw next to it another neuron. Like so. Another axon. And it'll end with some more axon terminals with terminal buttons. I don't know why my terminal buttons look like feet, but that's all right. That'll do. Okay, so let's kind of make sure we know the main parts even here. So these are going to be, coming off here, are going to be the dendrites. And the dendrites have on them receptor proteins. So dendrites with receptor proteins. That's what I've kind of drawn here as the little receptors. Then we have our long part here, which is going to be my axon. And then what we have down here is going to be my axon terminals. And we call the very end of it the terminal button. So axon, terminal, terminal buttons. Okay, so adding a few more things in. I don't think we named this yet. The soma. That's just the cell body that's going to be in charge of the actual kind of maintaining of the cell function. That's where the DNA is going to be. Okay, then I want to draw some myelin. And that would really be on both sides, like so. And same over here. We'll draw it as the cells. So the key is this is the myelin sheath. And that is made by Schwann cells. And they are going to wrap themselves around. So they are initially living cells that wrap themselves around and around the axon and eventually can basically push out the nucleus. And what they're left with is these layers of myelin sheath. We do want to know what the areas between them are called. So the areas between each little bit of myelin sheath are called nodes. So these are nodes of Ranvier. So nodes of Ranvier in between. Okay, so what I want to show here with this, so that should cover the parts of a neuron, is I want to clarify this difference between chemical signaling and electrical signaling. So when an action potential is sent down a axon, so it's going from positive to positive, and we are drawing it kind of skipping between the actual myelin sheath. So this process of transmission within a neuron is called electrical signaling. So that is going to be done with an electrical charge that moves along. So that's electrical signaling. We also know that that electrical signaling is called action potential or action potential propagation. So action potential propagation for the movement of the action potential down the axon. So within a neuron, that is electrical, so there is no chemical substance being passed along. It's just a charge. So the idea is similar to if you plug in a charger and you actually plug in your phone, it is not putting little chemicals into your phone. It is using electrical charges to actually do that. So that is electrical signaling. But between two neurons, there is the release of actual chemicals. So inside the terminal buttons, we have the little vesicles of neurotransmitters. They are going to get released into the synapse and then ultimately bind at a receptor site. So this is chemical signaling. So that does actually require a ligand, which is a neurotransmitter. So HL, that's our terminology. So within a neuron is electrical, between neurons in the synapse is chemical signaling. So that's just a little bit of context to look at this idea that there are two different things. So we're going to do first looking at the actual electrical signaling, and then we'll look at the chemical signaling next. So just a way of breaking it down into the two parts. Again, just so I have more space, I'm going to just keep erasing as I go, but you're just kind of continuing down on your page. So what I want to do now is look at electrical signaling. So we're going to say electrical signaling. And another way of saying electrical signaling is action potential propagation. So the spreading or the moving down of an action potential. Okay, so we are going to draw this in terms of the 4 steps. It's a little bit crowded because what I'm trying to show is that it's moving along each time. So I want to draw like essentially 3 sets of ion channels at every place so we can see the progression. So that is that added little bit of complexity in what I'm trying to do. So you can, if you want to kind of allocate your space, we're going to we want to use all the space you can on your page. So I want to draw 4 kind of axon sections, like so. Okay, and I can erase and open and shut mine easier than you can, so you may want to kind of wait till I finish and then kind of go back and do the channels at the end, or you can just follow along and erase if you can. So these are kind of my 4 steps involved in action potential propagation. So all 4 of these steps are happening. And we are really just primarily looking at what's going on at the beginning of the axon. So for each of these points, we're going to focus on what's happening right at the beginning of the axon, and then we'll show what's going on in the rest of the axon as well. So we're focusing on what's happening at the beginning. Okay, so again, I want to show that essentially the axon is a collection of sodium channels, potassium channels, and sodium-potassium pumps. So we're going to show that this is kind of like a series. So this is a sodium channel, followed by a potassium channel, followed by a sodium-potassium pump. And what I'm showing here in the first step at rest is all of the channels are closed. So like I said, I want to draw essentially 3 sets per axon because we're just doing that to show a progression. So there we go, and everything is closed right now. Now the channels are going to be on both sides. I'm just only drawing it on the top, but note that they would be down there as well. We're just limiting our drawing. So it would be coming in on both ends. So those are my sodium channels, potassium channels, and sodium-potassium pump. It'll be clear what they're doing as we go. So this axon right now is going to be at resting potential. So this step 1 is going to be resting potential. So let's look at where my ions are at resting potential. So at resting potential, we are going to see that my sodiums are all outside. So we're going to have lots of sodium all on the outside. So let's draw some good sodium on the outside. The exact number doesn't matter, we're just showing that. Meanwhile, my potassium is going to be on the inside. So technically, I probably, if you had the space, I would have you draw 3 sodiums to 2 potassiums, just because we're trying to make the idea that it has an overall negative charge. So it doesn't matter the exact number, but if you can, try to fit in more sodiums than potassiums, just because that will help us kind of understand this idea of relative charge. Okay, so we have sodium on the outside, potassium on the inside, everything is closed. And even though they're both positive ions, there is more positive sodium on the outside than there is positive potassium on the inside. So as a result, this is going to have a relatively negative charge. So the inside is going to be negative comparative to the outside. So the membrane potential, which is the relative charge, so membrane potential, which is going to be the relative charge, at this stage is negative 70 megavolts inside. So the inside of the axon is negative compared to the outside. So that is what is going on at resting potential. For the start of action potential, we're going to cover what starts an action potential more when we get to our chemical signaling. So right now we're just starting the action potential. We'll talk about the role of neurotransmitters in doing that. But the action potential is going to start with the opening of the first sodium channels. So my first sodium channels are going to open, and that is going to cause sodium to rush in only at this area. So that is what is going to start my action potential. We're going to make a note, the potassium channel still closed. The sodium-potassium pump inactive. And then this is where I'm showing you kind of the 3 stages, because I'm wanting to make the point that it's only beginning at the beginning of the axon. So they don't all open. So this sodium channel is closed, and the rest of the sodium remains on the outside. So it's only moving in at that one kind of initial place. Everything else is closed. Potassium is closed. Potassium is closed. And my sodium-potassium pump is currently inactive. Closed, inactive, whatever. Okay, so this one part of the axon is now really, really positive, or less negative. It's going to become positive. So because the sodium is rushing in and the potassium staying in, it's going to become positive from negative. So the influx of sodium is going to cause that positive charge. The rest of it is going to continue to have a negative charge. So it's only happening at that one area. So this is happening at the first chamber. We are calling this, if you're HL, you have to use the term depolarization. If you're SL, you can just say the sodium influx, so the sodium coming in or the change in the positive. That vocab term is not actually in the HL syllabus. But I'm going to put it down because I feel like it doesn't hurt. You have to understand what's going on. You just don't have to use the terminology for it. So depolarization is going to be the step. And what we want to be clear is that the effect of this depolarization is going to trigger the next sodium channels to open. So it is actually what is going to cause the propagation, which just means continuation. So down here, the second set of sodium ion channels is now going to open. So now it is going to spread along. So the next place on the axon is opening. So suddenly this middle section is going to polarize. Everything else in the middle section is the same. Okay, so that is what we mean by propagation. This triggers that. Once that has happened, once the sodium has come in and we're already moved the action potential along, that sodium channel is going to quickly shut, and in doing that is going to trap my sodium inside. In response, though, the voltage-gated potassium channel opens. So the sodium channel is actually linked to neural transmission initially, and then linked to essentially the spread through the action potential. But once that has happened, now the potassium channel next to it is going to open, and my potassium is going to go out. So my sodium is locked in, my potassium is going to go out. So now this initial section, once it's already spread the action potential, this initial section is going to become negative again. And the potassium channel is actually a little bit slower to shut than the sodium one. So it actually has a massive removal of lots of potassium, then evens out. So it has this kind of extreme removal of potassium compared to sodium influx. So it's actually going to become even more negative. SLs, you don't need to worry about that. HLs, you just need to understand why it dips on the oscilloscope. You don't really need to know why, just that when the potassium goes out, it actually becomes even more negative. So this idea that because of the potassium leaving, we now become negative again, this is called repolarization. So again, repolarization is specifically referring to what's going on right here. So it is referring to just the first set of channels. It is referring to the potassium leaving. Depolarization is referring to what's going on right here with my actual sodium channel. So this area is repolarizing at the same time that the next area along is depolarizing. Nothing has happened over here on my third set of channels, so we can draw them kind of still all shut down because nothing is happening to them at the moment. So they are all closed. So my potassium is still on the outside. Sorry, my sodium is still on the outside. My potassium is on the inside. And the charge is negative. Okay, let's look at then what's going to go on. And what I'm going to do is I'm going to go work backwards and then we'll work out what's going on here. This second section now has the positive charge. So this positive charge here is now going to trigger the opening of my last set of channels. So now I'm going to have my sodium influx all the way down here at the end. So at the very, very last place, I'm going to have my positive charge, my depolarization going on. Oh, sorry. Nah, you're still in. Too many things going on. And my sodium potassium pump is inactive. So if you just look at this positive, you can see it starts at the beginning, moves to the middle, moves to the end. So that's the propagation piece, the movement of the positive charge. At this middle section, because it has already spread it, it is now going to close, trap the sodium in, it is now going to be opening its potassium channels, and it is going to be in the process of repolarization. So my second section is repolarizing at this time. So now let's look at what's going on in the first section. So that's kind of our focus. The point of drawing the second ones is just to show the progression that is moving along. That doesn't happen to the whole axon at any one point. If you were to look at an axon at the beginning, you would see one area that's depolarizing, one area that's repolarizing, and so forth. So this is just showing this idea of progression. So if we look at my initial one, here it has depolarized, sodium came in. Here it has repolarized, the potassium went out. But everything's in the wrong place now. So where we left off, my channels were all closed. So my sodium channel closed and locked, my potassium channel closed and locked, but nothing's where we want it to be. So nothing's in the right place. So it's not set up to then do another action potential. So if it just stopped here, that neuron would be a bit stuck. It couldn't deliver another message. So now my sodium potassium pump is going to become activated. So this is going to become activated. Woohoo, it's moment in the sun. So what it's going to do is it's going to take the sodium and it's going to actually pull them out and return them to the outside. And then remember, for HL, you do actually have to know how the sodium potassium pump works. It's actually part of your B2, B2.1. And so it actually is going to have a shape change. So it's going to grab 3 sodiums. It's going to use ATP to and move. That's going to remove the 3 sodiums, pick up 2 potassiums, and then remove them and move them inside. So it is going to be a kind of code transporter concept because it is going to pull these out. So it's actually going to pull in every 2 potassiums for every 3 sodiums that leave. So that's just that little bit of extra detail. And we do want to remember that this process requires ATP. So this part of the process requires energy. So now it's going to have its negative charge and everything is where it's supposed to be. So that's the last step, and now we could start over. So now it's back to resting potential and it would be available to collect another message. So this last step is going to be return to resting potential. So all of these 4 steps are happening at each individual ion channel. So at each ion channel available, so if it is myelinated, it only happens at the free myelin channels, the ones that are in the nodes. If it's unmyelinated, it's happening at every single place along the axon. So at every single place along the axon, it's depolarizing, repolarizing, returning to rest. So all 3 of those things together make up a single action potential. And as that moves along, that's the propagation piece of it. So we're going to make a note that this uses ATP for the pump to begin where it started. So basically to restore the membrane potential to what it should be. So it wants to get it back to that negative 70. And not just to negative 70, but negative 70 with sodium on the outside, potassium on the inside. The only thing you can make a little note here for HL really is that it does slightly hyperpolarize when the actual potassium goes out. So slight hyperpolarization, which just means even more negative. Does occur. So again, you don't have to get into the nitty-gritties of the actual channels and why that happens, but when you look at it on oscilloscope, you will see that pattern. Okay, so that's really the most challenging part of this whole thing. And hopefully that didn't make it more confusing. If you're like, oh, the 3 channels just couldn't handle that, then just draw the first one. So in theory, you can just draw what's going on at the very first place. The purpose of the other ones was just to show this idea that the positive charge is moving along, and that essentially it's happening as soon as the first section depolarizes. The next section will start depolarizing while this one's repolarizing. So it's just to show a little bit of that real-time situation. Okay, so next thing we want to do that's still on this section is we want to look at the oscilloscope for HL, and then for SL we want to look a little bit at some of the things that impact the speed of this process. So kind of looking at the role of myelin, but a couple other things as well. So I'm actually going to do the SL part first, and then we'll do the HL part. Then there is more SL stuff when we get to the synapse, so just do a short little fast forward through the oscilloscope. So we'll look at like I said, 2 other things we need to pick up. So for HLs, we need to look at oscilloscope traces. And that's going to be HL only. So I'm just allocating some space for it. Okay, and then the other thing we need to look at is the speed of the action potential. So speed of action potentials. And in particular, we're really looking at the speed of the action potential propagation. So how quickly does it make its way down the axon? So it's not necessarily about what's going on at one channel, but the speed at which it spreads all the way down. So like I said, we'll come back to this. Let's hit the speed because that's an everybody thing. So there's 2 different factors that you have to know that it's influenced by. So one kind of random specific point is it's actually influenced by the diameter of the actual axon. So it is going to be increased by increasing, so positive correlation, axon diameter. often usually inversely correlated or negatively correlated with organism size. So it's really about the axon, not the organism. So just being clear on that. And there is a specific example that you could be given data on. So the giant squid axon. So they're actually unmyelinated but have a really, really thick diameter. So then the question is, why does that help? So the benefit of the thicker diameter is it actually allows for kind of more rapid ions flowing. So rapid ion flowing. So the ions kind of diffuse more quickly, and that actually speeds up the process. So the ability for there to just be more space for them, the ions to quickly get in and spread out and make that charge change is what causes that. It doesn't actually say you really have to account for why, just that you know that the giant squids have great fast neurons. Good for them. The next thing that is going to be increased by is that the speed is going to be increased by the presence of myelin. And you do have to be able to account for why that is the case. So what that means is that the actual action potential is only propagated between the nodes and not between each channel. So the action potentials propagate between the nodes. So they move through the myelin to the next set of nodes. So they're actually going to bypass some channels. Sorry, the sun's a bit, I was actually trying to have it no sun because I think that's better than, anyway, there's a streak of afternoon sun. So it enables it to bypass some of the channels that are covered up with the myelin, and that means less channels have to open, so it improves the speed. If you are HL, you need to know that this is called saltatory conduction. SLs, you don't need to know that term, but you still need to know the concept. So it's a little bit random that you don't have to know the word, but you just have to know what actually happens. The other thing that you need to also know about this is that it saves or uses less ATP. So the less channels that are opening, the less work on the ATP pumps, and therefore you're saving some energy as well. Okay, so those are the two things that are going to increase the speed. Okay, HLs only now. We're going to draw your oscilloscope trace. We're not going to draw it super giant. We'll draw like kind of a small one just to show the main points. Okay, so down here is going to be time, and it's usually given in nanoseconds. Random little skill thing I thought we would make a note of is it does say in the syllabus that you could be asked to use an oscilloscope to determine how many action potentials could happen in a second. So you could work out the total time and then work out how many of these therefore you could fit in a second. So we'll just make a note of that you could use this to actually determine the number of action potentials per second. So you're going to look at the idea that the nanosecond is 1,000th of a second. And so then essentially if it took like 100 for it to 100 nanoseconds to complete it, then you would only be able to do 10 per second. But if it only took like 50, then you could do 20 per second. So you could calculate that, just being careful to remember that nanosecond is 1/1000th of a second. Okay, then up here we're going to have the actual voltage. So this would be the voltage or the membrane potential, megavolts, like so. Okay, then you have to know the basic shape. So you would need to be able to draw the shape, which is that we're going to have it at rest. Then you might see this kind of starting and then come back down, and then finally it'll start, and then suddenly this rapid up, then rapid down, and then return to rest. So that rest should be the same. So that's the general idea of what you're going to see. On here, let's add this idea of threshold. So also I want to give a couple numbers. So right here at rest, this would be -70. That's going to be the resting potential. These ones here are what are called failed initiations. So these were too weak of stimuli. So they did not reach the number, the value they need. They did not depolarize enough to actually start an action potential. Easiest way to understand that is with sensory neurons. So there are sensory things that we never actually receive information from in our brain. And that's simply because the action potential, the stimulation to our sensory neurons is not strong enough. Okay, so the point that it has to reach to actually start the action potential, that is going to be threshold. And so it has to reach threshold, which is somewhere around -50 to 55. Well, let's put -50 will work. So -50 megavolts. So anything that's lower than that, that doesn't make enough of a change, isn't going to go through. So when we do the next section on the synapse, remember that can be related to the balance of inhibitory and excitatory neurotransmitters. So if they're both going at once, it can be this kind of balance out act. And it could be that you have enough excitatory, but the inhibitory ones are blocking the signaling, and that's why it doesn't reach. But it needs to hit that threshold point. Assuming it does reach threshold, it then very quickly goes up. So this is going to be depolarization. Depolarization always reaches +30. There is no change. This is an all or nothing event. So if it hits threshold, it will go up to +30. It won't be an extra good action potential that reaches +100. It's not going to be a sad little action potential, only reaches 15. Always 30. So it looks the same. So your brain can still differentiate based on how many action potentials it reaches, but it can't differentiate based on like the bigness of an action potential. So it is going to be a single kind of curve that's always the same. Then it will just as quickly repolarize. When it dips below rest, we call that hyperpolarization. And then it is going to return to rest. And the action potential is not done until it has returned to rest. So essentially the action potential itself is going to be everything from here, so from this point here through to when it returns. So if you were measuring the time, that is your actual action potential. So from the beginning of depolarization through to when it first is back at resting potential, that little section there is your action potential. So everything before that didn't reach threshold. Once it's returned to rest, you're good. So that's what you are looking for when you study an oscilloscope trace. Okay, so that is all things action potential. The last section is now to look at the actual chemical signaling. So what's going on at the synapse? Okay, so we're back to SL stuff as well. So hopefully SL, you've realized my board is blank and it is time for you to return to us. So now we're looking at chemical signaling. So now we are looking at what's going on in a synapse. So the main one that we're going to draw, so we're going to actually draw what's going on with an excitatory neurotransmitter, and then we're just going to cover what the difference is between that and inhibitory ones. So we're going to look at chemical signaling by excitatory neurotransmitters. And then we'll do a comparison for HL only to say how is this different if we were looking at an inhibitory one. So we'll pick up that piece of it afterwards. The specific one that you need to know is acetylcholine. Acetylcholine is involved in lots of different things, but the main context that we're looking at it is for muscle contraction. So it's involved in between neurons, but also then it reaches the neuromuscular junction, and it's included because it's something that we've done in another context. So it's always good to have kind of a bit of both because we use it in motility for HL. Okay, so we're gonna draw a little synapse here. I just need a little bit of room at the end for a last couple, the sun's driving me crazy, for a last couple comments for HL. And then over here, we'll have a couple other HL comments as well. So SLs, all you really need is just this synapse, so you can draw big. HLs, we need to add a few more things. So we'll draw kind of our zoomed in now. So here's my terminal button. And then here's my dendrite of the next one. Okay, so a couple of other terminology things that I didn't use last time, but we do want to add. So we're going to add that this is called the presynaptic neuron. That's the one whose axon terminals or terminal buttons are facing the synapse. This is going to be my postsynaptic neuron. And that is going to be the one whose dendrites are facing. So presynaptic, postsynaptic. I'm just going to try something to see if this makes it better or worse. I tried to see if I could get rid of the line, but I cannot get rid of the line. So we're just going to embrace the line. Okay, so now I just have a lot of light in my face. So those are some additional terminology that we need to add. Okay, so we're going to say what's happening when this actually delivers some neurotransmitters. So that's going to start with an action potential arriving. So the action potential arrives at the end of the axon. That action potential's positive charge is going to cause for the opening of voltage-gated calcium channels. So this is a voltage-gated calcium channel. And so as the action potential arrives, this is going to open, and that is going to cause an influx of calcium. The calcium is going to come in and it is going to bind to these vesicles that were already sitting in here. So that's a vesicle, and inside the vesicle is the neurotransmitter or the acetylcholine. So my acetylcholine is sitting in this little vesicle. When that binds, that is going to move my actual vesicle to the synapse or to the end of the axon terminal, and it's going to merge with the actual membrane because vesicles and membranes are both made of phospholipids, so easy for it to merge. And that is going to dump my acetylcholine into the actual space, which is the synapse. We can also have a special name for just the space, and we can call it the synaptic cleft. So that's referring to the actual space. So there's my synaptic cleft. Okay, so that's essentially what's going on in my presynaptic neuron. So action potential arrives, opens the calcium channels. Calcium channels open, allow calcium in. When the calcium comes in, it merges with the vesicle. The vesicle then moves to the membrane, and then it essentially emits the neurotransmitters by exocytosis. Then let's look at what's happening over here on my postsynaptic neuron on the dendrites. So I'm going to draw essentially a receptor that does not have any acetylcholine. So HLs, we did this a little bit more in our last topic. So that one is going to be closed. What's going to happen when acetylcholine binds is it actually is going to change shape and create a channel. So when acetylcholine binds, here's my acetylcholine binding. What that did is that caused the shape change. So that actually caused this to change in shape. And it now created an open ion channel. That open ion channel is now going to allow for sodium, which had been sitting outside, to enter. So now the sodium enters the actual neuron. Assuming that enough sodium enters to reach threshold, so this is going to affect the membrane potential. So this is going to cause excitatory membrane potential. So it's going to excite the membrane. And as long as it is enough to reach threshold, that is then going to cause the opening of the first set of sodium channels. So then the sodium coming in through the actual channel that was opened by acetylcholine is going to ultimately, as long as there was enough excitation, lead to the first set of channels opening. Then my sodium is going to start entering, and then the next action potential is going to begin. So that is going to depolarize, and then the next action potential begins. So the action potential is initiated. So again, the only little caveat here, this is probably more of an HL thing than an SL thing, is that there has to be enough for threshold to be reached. So it has to be enough excitation to cause that to happen. So presynaptic neuron releases the neurotransmitter into the synaptic cleft. Postsynaptic neuron has the actual receptors. When acetylcholine binds, it causes a shape change, which creates an ion channel, which allows the sodium in, which causes an excitatory membrane potential, so changes the membrane potential. As long as that is enough to reach threshold, it will open the sodium channels in the axon and lead to the action potential beginning. Last little thing we want to add in here is that if we were to just leave acetylcholine in, so once it's done that, it will detach from the receptor because it doesn't enter the cell because again, it is not hydrophobic, so it can't cross in, so it will detach. If it just sat in the synapse, it would keep triggering more and more action potentials, which would not be ideal. So here in my synapse, I'm also going to have an enzyme that is going to break down the acetylcholine so that it doesn't sit for too long in the actual place, and it will recycle the choline back into the presynaptic neuron. So it will take this, it will break it down, and then it will recycle the parts back inside so it can be reused. So it's a little recycling machine that enables us to reuse the parts of acetylcholine and also gets it out of the synapse so it doesn't just starts causing more and more action potentials. That enzyme is called acetylcholinesterase. I think that's right. I maybe had an extra cholinesterase. Doesn't have a T in it. Again, so that is the enzyme that is going to break down and recycle the actual acetylcholine. And that prevents it from staying in the actual synapse. So it breaks down and recycles it, and in doing that, it removes it from synapse. And that's a good thing because that means that we only have muscle contraction every time our brain has commanded it, and we don't end up with this excessive response whereby our brain tells us to do it once and then we're just doing it over and over and over. So that enzyme is going to sit there in that. Okay, so that's everything for SLs, so you guys are good. HLs, we need to add a couple more things that we're gonna add to this to kind of add the remaining little bits and pieces. So down here, supposed to be down here, we're gonna add a little bit on exogenous chemicals, and then we need to also say what if it was instead an inhibitory neurotransmitter instead of an excitatory one. So we'll do the inhibitory first, and then we'll pick up on that. So again, this is an excitatory neurotransmitter, and what we showed is that it causes the action potential. Now we're going to look at what if instead it was inhibitory neurotransmitters. So we're going to look at how that would change. And again, I'm sorry, I know there's like a line. I can't really do anything about it. It's getting bigger as the afternoon comes upon us. So for the inhibitory neurotransmitter, I want to start with what's the same. So what is the same is that it will also bind to the receptor. So we want to be clear on that because there is a tendency to get confused and think that they're the opposite, but they actually are very similar. So it will also bind to the receptor and activates it, activating a shape change. And that shape change will also opening an ion channel. So a lot of it is the same. So the way it all works is essentially the same, but here's the difference. We clearly showed here that what was allowed in was sodium, which is a positive ion, which caused a positive excitatory membrane potential. So it made the membrane more positive. But if it's an inhibitory neurotransmitter, it is instead going to allow chlorine ions. So chlorine ions enter instead. And the chloride ions that enter instead, because of their negative charge, they actually cause an inhibitory neurotransmitter or membrane potential, meaning they further decrease the membrane potential. So further polarizing the membrane. So the term we use for that is that they actually hyperpolarize. So they are going to hyperpolarize the membrane, causing it to become more negative. Because it is more negative, that actually means it's even less likely for it to respond to an action potential in the future. So it is going to become hyperpolarized, and that hyperpolarization is going to prevent action potentials. So not only is it not itself going to cause an action potential, but it's going to mean that the next action potential is even harder to accomplish. So it is going to prevent the future action potentials. So inhibitory neurotransmitters can be really useful because they basically slow down the oversensing of messages to your brain, so they can cause kind of a calming effect whereby they actually stop your brain from sending excessive numbers of messages. So they can be really useful because they actually kind of decrease the membrane potential and make it even less likely for other excitatory neurotransmitters to reach threshold. So that's the role of the inhibitory neurotransmitters. So again, you just need like the world's tiniest amount of space down here. So we just need to add a couple of extra HL things. All right, so the last little thing is there are two exogenous chemicals you need to know about. So there's two chemicals that an individual can be exposed to that can interfere in this area. So endogenous chemicals are chemicals our body makes. So neurotransmitters are endogenous chemicals that we make ourselves. But you can also have exogenous chemicals, which are chemicals that we insert in our body. So that's kind of our last little piece, is to look at exogenous chemicals that impact the synapse. We did a bit more and drew these in class, but for now I'm just going to list the two that you need to know and the kind of bare basics of what they actually do. So the first one that is named that you need to know is the neonicotinoids. So these are actually the man-made compounds that are used in insecticides, and they block acetylcholine receptors. So they're used in insecticides. They're going to block acetylcholine receptors in insects. And they also, interestingly, they don't actually start, so they block it, but they do not actually cause action potential. So they're going to block the receptor, but they do not activate it. So they actually prevent acetylcholine from binding, and therefore there's no action potentials. So it cannot initiate these action potentials. It also is shape incompatible to acetylcholinesterase, so it cannot be broken down. So it just stays in the synapse. So the acetylcholine would be broken down, the actual block stays there, and so essentially it's just unable to initiate any sort of action potential, and it's going to cause paralysis. So inability to create any movement, and then paralysis is going to cause death because that's going to prevent movement of heart muscle and things like that as well. So that's like a man-made one that has been created in order to essentially limit the neurotransmission of the insect. The other one that you need to know is cocaine. Cocaine does not actually bind to the receptor. It actually blocks the reuptake pump. So it blocks dopamine reuptake pumps. So by blocking the dopamine reuptake pumps, it actually keeps dopamine in the synapse. Therefore, it kind of causes repeated activation. And that repeated activation means instead of a momentary pleasure feeling, a feeling of reinforcement, it can cause that kind of excess feeling of lots of that at once and cause kind of a euphoria. And we talked a bit more in class about how that's then problematic because what ends up happening is the dopamine receptors get desensitized to having so much dopamine there all the time, and they stop responding well to it. So then without the cocaine, people stop responding well to their natural dopamine. So there's lots of issues associated with that, but that's essentially how it causes that sensation. It does not introduce a new chemical, it just causes you to use your chemicals more repeatedly. Okay, and then the last little, this is just a tiny point, but I just thought I would include it because I try to make sure I mention every little dot point. So everything that we've been doing so far has been this kind of zoomed in. That's a little magnifying glass. So what we've been doing is we've been zooming in on one small process and understanding it in detail. So we're looking at a single process and trying to understand it in detail. And this idea of looking at one, let me just change that word small to single. So we're looking at one part of a complex thing and trying to understand it really well. So we're trying to say, okay, let's look really clearly at dopamine and try to understand how it works, and then try and say, well, now we understand addiction. So when we do that, we're falling into what's called reductionism. If you do psychology, you would know this term quite a bit more. So it's a kind of a common complaint. So again, if I was talking about addiction, and instead of talking about upbringing and stressors and all this, I was like, it's just dopamine related, too much dopamine in response to something, and that's why someone has addiction. I'm doing reductionism because I'm taking a really complex issue and I'm actually breaking it down into a really simple process, which is helpful, but it loses some of the big picture. So instead of doing reductionism, it's also important to sometimes zoom out and look at the whole, and look at interactions and kind of how different things work together. And when we do that and we look at the more complex things, then what we can see is these emergent properties. So emergent properties are things that we can't necessarily explain by looking at a single process. But when the processes all come together, you end up with this like the whole is greater than the sum of the parts. So break it all down, put it together, and yet there's something bigger. So the example they want you to know for that is consciousness, which we already looked at a little bit when we did C3. So this is just a reminder of that. But essentially, like if we study the interactions of neurons, we can understand a lot, but we don't really understand what enables us to be aware of things and what causes metacognition and thinking about thinking and some of the more complex ways that we understand and are aware of the world, we can't really account for well with neurotransmitters. So that's just saying that, inevitably looking at individual neurons is a somewhat reductionist thing, and it is important to recognize that there is more complexity to how our brains operate and how our neurons send messages that we can't necessarily understand on that microscopic level. All right, so that's all for neural signaling. Sorry about the sun.